Hi, everyone. Welcome to the Live Longer World podcast. Through this podcast, I have conversations with scientists, biotech entrepreneurs, investors, and other advocates that are transforming the field of longevity science. We have conversations on how you can slow aging, reverse aging, and maximize longevity now and in the future. If you are new here, my name is Astha Jain and I am an advocate for longevity science. Besides this podcast, I also have a newsletter where I often discuss longevity lifestyle hacks and some basics of longevity science. On my YouTube, I also create some other videos talking about aging as well as longevity lifestyle tips. If you wish to listen to this podcast on other platforms, you can find it on Apple Podcasts, Spotify, or whichever other medium you prefer. All right, that's it for now. And with that, let's get started with today's episode.
My guest today is Reason, the founder and CEO of Repair Biotechnologies. Repair Biotechnologies is a biotech company focused on developing therapies that treat medical conditions arising due to the accumulation of excessive cholesterol. They've developed a unique cholesterol degrading platform to reverse the damage that leads to heart disease and atherosclerosis, which kills 5 million people every year. Reason is perhaps most well known for his popular blog Fight Aging, where he distills the latest research in aging science. For anyone who's looking to stay informed in this field of longevity research, I highly recommend you checking it out. All right, so in today's conversation, we discuss how to think of aging simply. What are the differences between slowing and reversing aging, NAD plus boosters and their effectiveness, slowing aging with exercise and caloric restriction, reversing aging using senolytic therapies, how his company is working on a gene therapy to reverse atherosclerosis and cardiovascular disease, how one can get involved in the aging space, be it through investing or other means, and much, much more. There is one thing to note. We discuss a fair bit on cellular senescence and senolytic therapies. So for those who are unfamiliar with the concept, cellular senescence is a process in which cells lose their ability to divide, but they refuse to die. And when these senescent cells don't get cleared away, they release harmful substances and damage the cells around them too. So think of it as one bad apple that makes the other apples become rotten as well. And it is well known that senescent cells accumulate with age, they contribute to inflammation and aging itself. And what Senolytics is doing is that it's a class of drugs that can actually selectively clear away these senescent cells. And they've actually shown a lot of promise in delaying and preventing age-related diseases. All right, that's all you need to know. And with that, let's dive right into the conversation with Reason.
Great. So, hi, Reason, and thank you so much for being on the Live Longer World show. I really appreciate your time.
Always happy to be around. It's good to talk.
So as I was saying before, I do love your blog, and I hope a lot more people read it, Fight Aging. It really does a great job in advocacy and talking about aging. And I think the point I'd like to start with is just talking about aging very simply to the general public. You do a beautiful job in explaining aging as damage accumulation and something that's increasing your risk of death with the years to come. I'd love it if you could elaborate on that point and just explain aging to someone who's probably never thought about it in terms of aging as a disease.
So sure, I think like the rusting car is a good way to think about it. If you leave a car out in the field for long enough, it will rust and it will fall apart. And because a car is kind of complicated in its structure, as it rusts, it could fall apart in all sorts of different ways. Well, will the roof cave in first? Will the door fall off? Any one of those things stops it being a viable car to drive around in. But it's very unclear as to which of those things will happen first. Rust is very, very simple. It's just a very simple little chemical process. But if you start rusting a complicated piece of metal, it will fall apart in complicated ways. And aging, if you want to think of it very, very simply, it's just a set of rusts. We rust if you want to take the analogy, we accumulate sort of damage and dysfunction as a result of very simple processes that go wrong and create problems that accumulate over time. And aging is really only complicated because we are complicated. If you create a simple form of damage in a complicated thing, like a person, then the person will fall apart in all sorts of weird and strange ways, and everyone falls apart a little bit the same, but a little bit differently. Different things happen earlier. Some people get cardiovascular disease, some people get cancer, some people get neurodegenerative disease, and that's what kills them first. But it's all the result of the same underlying simple processes that are causing damage. And we evolved this way simply because evolution really wants you to succeed early in life. The organisms that win the evolutionary battle for who gets to survive are the ones that reproduce the earliest they could within the constraints of your environment. And therefore, evolution likes to front load. You get systems that are made for short-term use, like your immune system. Your immune system works great out of the gate. Your adaptive immune system, the one that remembers problems it encounters, but it keeps having to assign space to its memory, and eventually it'll run out. And that's kind of a problem. It never evolved to carry around for the long term because evolution only really cares about, did you successfully have offspring before you were 30? Which is roughly what your grandparents care about too, but that's the way it goes. So yes, aging is just damage, and it's very simple in comparison to what the damage does, which is very complicated. So when you look at the research community struggling to try to explain little details about aging, how it actually works all the way along, the reason that they're struggling is because we still don't fully understand human cellular metabolism in all of its details. So it's rather hard to explain what happens in a complex system if you don't actually understand the system fully. And researchers write grants for things they can write papers on. So they're very interested in explaining aging, but we don't really need to explain aging. We actually have a pretty good understanding of what these fundamental forms of damage are, a good enough understanding to go out and try to fix them. And frankly, the best way to figure out, what does aging do while it moves forward in this incredibly complicated system is to take an animal where something hasn't been fixed and take an animal where something has been repaired or fixed and see what the differences are. And we do that for things like senescent cell clearance. Where senescent cells accumulate with age and they spit out all sorts of gunk and inflammatory signals and mess up your body actively. And animals in which you get rid of senescent cells have quite profound rejuvenation. A lot of their bodily systems are given leave to go back and try and get themselves back to a normal operating status, and that can be really quite impressive. Let's, for example, take cardiac hypertrophy. When you get old, your heart starts to expand. Your heart muscle expands and weakens at the same time, and it contributes to heart failure. And that's a pretty horrible thing. And you wouldn't think that would actually reverse. But if you go clear senescent cells in rats, yes, it does. It actually, the hypertrophy of the heart actually starts to reverse, and the heart goes back to the way it should be and pumps more strongly. So there's tremendous opportunity here just by looking at comparatively simple forms of damage in the body and focusing on that and leaving the question of, okay, great, but how does this damage really actually then cause all sorts of problems in your body over time? Leave that as a secondary question. Go fix the damage first. It will make it so much easier to figure out, if you really want to know at that point, what happens afterwards. But equally, tuberculosis is pretty much a solved problem. Does anybody out there really, really care about how tuberculosis progresses exactly in the body beyond what we already know? Ditto for smallpox. Do we really want to be out there researching exactly how smallpox, causes harm and dysfunction? No, nobody cares, because smallpox is cured and done with. And the same will be true of tuberculosis one day. The focus needs to be on fixing the causes.
Yeah, I think those are really great points. I think something you mentioned in the beginning was, rust it seems pretty simple on the car on the outside, but there are these complex chemical reactions that are happening, which could be similar to, just wrinkles start showing up or your knees start hurting, but then there's all this damage that's actually accumulating inside your body with age. Similar to how the immune system, we see how COVID impacts older people a lot more than younger people. And that's probably also something to do with immunity and just evolution, as you mentioned. But I think a couple of things you mentioned at the end were senescent cells and rejuvenation, for example. And you lay out the distinction really well between slowing aging versus actually reversing aging and rejuvenation therapies. And honestly, I hadn't thought about that distinction before that, before you wrote about it. And obviously, like tiny lifestyle changes, like, just eating better, caloric restriction, exercise, all fall in the category of slowing aging. But then there are also these NAD precursors or sirtuins, for example, and you put that in the category of slowing aging too. And I would love it if you could, one, explain the distinction between what slowing aging versus reversing aging actually is. And also, do you think some of these precursors or supplements are the hype around it is unwarranted in some ways?
Well, hype is almost always unwarranted in the industry. Sad to say, then they're in the business of selling lies and hope mostly. So, slowing aging and reversing aging sort of blur into one another in the middle. If you have a way to reverse aging just a little bit, and you do it repeatedly, then really that's just slowing aging. But equally, and by reversing aging, I specifically mean repair some damage. So if you can go repair just a little bit of damage, not enough to get back to where you were, but just enough to slow the accumulation down, that's roughly slowing aging. So bad rejuvenation therapies will slow aging. So equally, on the other hand, if you have a way to compensate, not repair the damage, but to somehow compensate for it, by forcing cells to behave as if the damage didn't exist, or by telling cells to just act better and be more useful despite damage, that will generally slow aging. But compensation is hard to do for the long term, and it falls off in effectiveness very rapidly. Things like exercise and calorie restriction, they're a very little bit of bad rejuvenation mixed in with a bunch of compensation. They're a little bit of both. They slow aging because they're preventing some cells from becoming senescent. They're making you a little more resilient to some forms of damage. They're slowing the accumulation of some other forms of damage. But I don't think anybody should be jumping up and down and saying exercise is rejuvenation. Even if you take a sedentary 60-year-old and you put them on a treadmill for a couple of months, they start looking a lot better. Right. But I don't think we can sit down and call that rejuvenation. That's just ending neglect. So slowing aging, really, I think most of what I would refer to slowing aging is where you're trying to do things where you improve cellular metabolism without necessarily attacking the damage that is causing cellular metabolism to become dysfunctional. So you picked up the example of NAD plus precursors. So NAD plus is an essential molecule in the operation of the mitochondria in your cells to provide power to your cells. Mitochondria package up an energy store molecule called ATP, and your cell needs ATP to do anything. So with age, your mitochondria become worse at producing ATP for a whole range of different reasons. And one of those reasons at the later stage is that you run out of NAD plus, you get less of it. And this is because of a whole bunch of different things that lead to the situation in which some production and some recycling and some other chemical reactions are not running as well as they should. But that's all way downstream of the causes of aging. It's a knock-on effect of a knock-on effect of a knock-on effect. But you can interfere with that by delivering some of the necessary chemicals that are not being produced in large enough numbers in the aged body, and try to just shortcut the later stages of this problem just for NAD plus. Of course, that doesn't change any of the other myriad things that have gone wrong with your mitochondria. It just changes the NAD plus problem. But that has a large enough effect in mice, at least, to be vaguely interesting. In humans, it's sort of an open question as to is this actually any better than exercise? There's actually a very long history of upregulating NAD plus going back 100 years or so, though they didn't know they were upregulating NAD plus until very recently. This is all high dose niacin is a very long tried, and there's sort of, it's very mixed. Does it do anything? There's very few good clinical trials that show any sort of reliable benefits. So you can absolutely take a human and upregulate NAD plus in their mitochondria and get the chemical results that say, yep, I increased NAD plus by 100%, which, by the way, you can do by going on an exercise program more easily. So, that you get the same problem with this sort of exercise and let's say any sort of calorie restriction mimetic drug, where they say you're forcing the nutrient sensing mechanisms to think that there's less food going on, so the cells suddenly get much more uptight about recycling and being careful and ramping back their protein production and generally tidying up the house. So you get an improvement, but again, you could go out and exercise and you get much the same thing, or eat less and you get much the same thing, except more comprehensively, because the drugs only touch a little part of the system. So this
what if you're combining the two, though? What if you're exercising and taking these supplements? Is that, do you see better, more effect?
You get some overlapping benefit, but equally it's still not as good as if you just ate less and exercised more. So, there's an awful lot of effort going into selling drugs to people who don't really want to go out and exercise. Right. But this isn't going to change the shape of a human lifespan. We know what calorie restriction does in humans. We know what exercise does in humans. You can't exercise your way reliably to living to 90 years of age. The fittest people, half of them are dead by the time you get to 90 in the present environment of what a 90-year-old has lived through. So it's not the way to go. These things are not interesting. Upregulating stress responses like calorie restriction mimetics, improving mitophagy in aging cells, the way it's being done right now, it's not a good enough approach. It's not a big enough benefit to be interesting. If you can't do much, much, much better than exercise, then why are you even bothering with this line of development? That's my take on these things. We need much better approaches, and to do that, you have to have things like clearing senescent cells or breaking crosslinks or all the other approaches that are sort of under development in early stages in the biotech community.
Okay. So let's discuss those, and then the rejuvenation therapies and clearing senescent cells. I guess I want to touch upon like funding for those and narratives for those, but maybe before that, because senescent cells seems to be quite promising already, just the area of Senolytics. What is the latest development in that field? What is the progress in terms of clinical trials or drugs, or can people start using it? What is happening there?
So Senolytics is the general name for drugs that kill senescent cells. It's a subdivision of the broader development of therapies that we'll call Senotherapeutics, which also covers efforts to prevent cells from becoming senescent and efforts to make senescent cells less horrible if they are going to be senescent. I think that the prevention has some promise in the sense that mTOR inhibitors do seem to prevent cells become senescent in enough of to a big enough degree to perhaps be interesting and competitive if it works out. There's not, there isn't the weight of evidence yet to say that this is actually real. But if you could find other things that can, let's say, halve the number of senescent cells in your body if you take it over 6 months, just by affecting the turnover. Senescent cell accumulation isn't actually, by the look of it, by the way, a matter of getting these senescent cells and they just sit there forever. It appears to be a turnover problem. You generate more of them because your body is more damaged and cells are more unhappy than you do in youth, and you get rid of them less effectively because your immune system is becoming dysfunctional. So if you could affect the turnover on either side, maybe you're good, but there isn't much. The work on destroying them is much more advanced than the work on preventing cells from becoming senescent. And there's always this fear that if you're preventing cells from becoming senescent, you're actually sometimes going to increase the risk of cancer because some of those cells should have become senescent because they were damaged. Senescence is an anti-cancer mechanism at root, one that goes out of control and becomes wild when you get too old. So Senolytics as an industry, which is what I was originally going to say before I sidebarred myself there, Senolytics as an industry is very, very unusual and very interesting, because the first drug combination that was developed in animals is actually really, really good. And nobody's yet done much better than that, where they've published data. So you have an industry where a drug, dasatinib, combined with a supplement, quercetin, is actually as good as whatever they're rolling out of the drug development pipeline. So it should be interesting to see how that pans out, because the first thing that's going to happen as soon as the Mayo Clinic finishes up their dasatinib trials in a way that's compelling, you're going to see enormous off-label use everywhere. And in fact, you're already seeing significant off-label use of dasatinib and quercetin, and a community of people growing who are using these drugs and claiming good effects from them in terms of, addressing their age-related concerns. So that's going to be a bit of a problem for all these people who want to charge you, $10,000 a year for whatever senolytic drug they're putting out. There's a tremendous diversity of those drugs being developed. There's gene therapies, there's immunotherapies. Various senolytics are under development. The one that will win is prodrugs, I think, because prodrugs have the advantage of what they do is they take a chemotherapeutics such as navitoclax or dasatinib. That is dasatinib's good because it's a pretty mild chemotherapeutic and a very good senolytic, and you don't have to take it very often. Navitoclax you have to take a lot more of, and it's a really ugly drug because it has very ugly side effects. But if you join it onto a compound that only gets removed in senescent cells, you get a harmless compound that does nothing to most of the cells of your body, and you can dose it up at large levels so that it really kills a lot of senescent cells, and it's cheap. So I think they'll win in terms of effectiveness, but will they be able to displace dasatinib and quercetin? Because dasatinib and quercetin is pretty good for a first outing, and it's an existing drug. It's manufactured in large amounts. It's already FDA approved, so anybody could use it off label, and people are. It's a pretty fascinating industry.
Are there any known side effects at all? And also, what age do you think people can start taking it? Like, if I'm in my 20s, should I be taking these?
That's a really interesting question. People don't know. So there's a lot of conflicting evidence that suggests that maybe we should be clearing senescent cells younger in life. But equally, it seems more likely that senescent cells only become important once you get to the age at which your cancer risk is high. That's an unresolved question. What is needed is a good set of data showing what is the senescent cell burden in all the tissues of the human body at a given age, until we can start saying, should you actually go in and take senescent cells? I mean, in fact, in general, people don't really know which of the causes of aging are important when you go from, let's say, 20 to 40. We have no idea. What are these things that happen to your skin and your brain and your gut microbiome? Why are they happening in the 20 to 40 range? And, who knows? The only way to find out really is to start repairing things and see what happens. But in the balance of, should I take a chemotherapeutic every now and again? If you're in your 20s, that probably doesn't meet the risk reward benefit. And then,
Right.
it adjusts as you get older.
All right. But what about fasting, actually, longer term fast? Does that also clear away senescent cells, or is that not as good as taking some of these off-label drugs?
It falls under prevention. I mean, if you can do something over 6 months that reduces your senescent cell burden by half, then taking Senolytics does about the same thing in a few days.
Yeah.
And it's not at all clear. Obviously, fasting does what fasting does. You can't fast to reverse your cardiac hypertrophy. You can't fast your way out of having atherosclerosis. At some point, these tools fail. They're largely preventative. But Senolytics clearly actually does quite dramatically reverse conditions in later life, at least in mice, and we will see whether it does that in humans. The Mayo Clinic is starting a trial for Alzheimer's disease because senescent supporting cells in the brain are, judging from the animal models, very important in producing neuroinflammation and neurodegeneration. And if you remove them in the animal models, things get a lot better. So let's say 5 years from now at the outside, we'll probably know whether or not the best Alzheimer's drug of the next 15 years is going to be dasatinib. It's quite possible.
Interesting. I'm very curious to see what happens in that field then. Shifting gears to your company a bit. So before you started Repair Biotechnologies, you were, I believe, angel investing in a lot of longevity aging startups and also writing a blog, obviously. And then you decided to start this company. I believe the focus at the beginning was on the thymus and repairing the immune system. And it seems like now you've shifted gears a little bit to cholesterol degrading. I'm curious as to why you shifted focus, if that's the case, and like what you learned about the thymus and immune system, and then later we can go into the work you're doing right now.
It's the tyranny of funding is the answer. When you start a company, if you have multiple projects, eventually you're only going to be doing one of them, and it's whichever one of them does better the fastest than the other one. You end up reinforcing success. Investors really like you to focus down. So eventually, the after a year or two, the cholesterol degrading approach just was producing amazing data in comparison to our other work. So we just kept shifting resources to that one, and eventually that's where it started. That's where it came to. I think that attempting to prevent the quarter of humanity that dies from atherosclerosis is a big enough job for now for this. Though I would certainly like to get back to immune aging, which is a very important topic, and it's broadly very influential on near every aspect of the operation of your body as you age. The immune system doesn't just defend against pathogens, it's also responsible for killing cancerous cells, and tissue maintenance in general is intimately connected with the function of the immune system. So, if you want to not be frail and live longer, having an immune system that functions in a useful fashion for as long as possible is very important. And at the point at which you're 50, that stops happening. If you're 50, you're coasting on fumes at this point, because your thymus has involuted and you're not really generating a large supply of T cells anymore for your adaptive immune system. So that you have about a 10, 20-year lifespan on those existing cells as they just replicate to keep themselves going, but they become increasingly sort of cranky and misconfigured and broken in the absence of sufficient reinforcements. So if you could just regenerate the thymus and replenish all of those cells, it's a very important component of immune aging. There are others, of course. You have to fix the bone marrow cells that are responsible for generating all immune cells, and you kind of have to clear out the cells that have become broken and problematic as well. But these are all incrementally, you should get benefits from doing each of these things. It's a very important project, and I'd like to see more work conducted in this direction. Ultimately, sometimes that means you have to do it yourself.
So if someone is working in this area of the immune
Oh, yeah.
system aging, can you talk about what you learned from early experiments there, like just your learnings and findings there, if it could be helpful or useful to
On
share?
which side? The immune side? It's
On
easier
the
to talk
side.
about the. I'm sorry.
Sorry, yes, on the immune side.
So I think what we learned is that the thymus is a very hard target. It's tough to work with, and it's very hard to find a delivery system that's going to get your therapeutic to the thymus without having too much of it going elsewhere. I believe that there are certainly ways around that, and that is the way I'd look at that. There are other people working on approaches to thymic regeneration. There's Intervene Immune, who use a growth hormone-based approach, which of course has its own downsides, though it appears modestly to work given their initial data. There's also a number of other groups that are taking a more cell regenerative medicine approach based on producing tissues that they can implant. And there's a variety of other things at very early stages where people are trying to find small molecules that selectively interfere in the thymic regulation of growth without interfering elsewhere. But I think that's probably a bit of a tall order because the thymus does share vital mechanisms with some other parts of the body. So I think it's going to be challenging to produce a small molecule drug that can provoke thymus growth without actually also doing bad things somewhere else.
I see. Okay, that's helpful. So atherosclerosis, a new project, cholesterol degrading. Can you talk a bit more about that and what you're trying to do there? And it seems like you also have promising results in mice so far, so would be great to hear that.
So atherosclerosis is primarily a condition in which the macrophage cells in your body become dysfunctional. These macrophage cells are innate immune cells. They just come straight out of your bone marrow and other locations, and they usually hang out in the spleen as monocytes, and then go into your bloodstream to go out and find problems. And when they find an area of your blood vessel wall that's inflamed or damaged, they dive in there, become macrophages, and start to repair it. And the major issue in the body in this context is cholesterol. Cholesterol is needed everywhere in your body. The cells use it to make up their membranes. Cholesterol is also not really destroyed locally. Cells have to go fetch cholesterol by picking it up from the environment. And when they have too much of it, they pass it back into the environment. They don't really have the option to manufacture it or get rid of it locally. It's expensive. So it's made, for the first approximation, it's made in the liver. Then it gets put into the bloodstream and carried around on LDL particles, and cells ingest these LDL particles as needed. And if they have too much cholesterol, they give it back to HDL particles that take it back to the bloodstream and send it back to the liver. And this huge Rube Goldberg system evolved probably because cholesterol is so abundant in the body that you can't evolve a way to just break it down without killing cells, because cells need cholesterol for their membranes. So what happens is you get too much cholesterol or oxidized cholesterol in the blood vessel walls, and that annoys the blood vessel wall cells, and they call for help. You get macrophages coming in, and they try to clean it up. And this works great when you're young. You don't get atherosclerosis as a young person. But later in life, you get these areas where it runs out of control. The macrophages come in, they try to clean it up, they get overwhelmed and die. And they contribute to this growing mass of fat in your blood vessel wall that just keeps adding more and more fat, and it's constantly calling for more macrophages to come in and try to clean it up, and they come in and they try to clean it up and they fail and they die, and this just continues as a feedback loop. So everybody so far in the research and development community has settled on, well, let's try to lower the amount of cholesterol in the bloodstream as a way to do this, and every single drug so far deployed does this in one way or another. But if you have an established area of plaque, this sort of fatty macrophage graveyard full of gunk that's just going to kill more macrophages, lowering bloodstream cholesterol really doesn't help that much. It just slows things down a little bit. You get about a 20% reduction in mortality at best. And in this environment where everybody and their dog is taking statins to lower their blood cholesterol, it still kills 25% of everybody. So we need to do better than that. And the doing better than that is, let's go get rid of the cholesterol that's in the plaque itself. That is the problem. And the way you do that really is by making the macrophages resistant to that environment in some way, shape, or form. And our approach is to genetically engineer the macrophages to give them the ability to break down cholesterol locally into a safe byproduct. So that enables the macrophages to, in principle, just continue to operate as they would in youth. And it doesn't matter how much plaque you have, given enough macrophages, they will break it down and get rid of it and gradually return the blood vessels to normal. So, any approach that made macrophages completely invulnerable to their environment would work. And obviously that getting that approach is kind of troubling. There's some things you can do that seem to work decently well in mice, but we think our approach is sufficiently comprehensive and direct that we don't have to worry about any of the fiddly problems about differences between mice and humans, or problems with metabolism that occur in some of those other approaches. And as you mentioned, our approach works really well in mice. We delivered an AAV gene therapy to mice, and with a pretty high dose. And it was perfectly safe, very well tolerated, and halved the amount of lipids in unstable plaque in these mice. And if you can get rid of the cholesterol that's making soft, unstable plaque soft and unstable, then if you get rid of half of that, then you've roughly halved the mortality risk due to those plaques rupturing and then causing a stroke or a heart attack. And that's what we do in a nutshell. And we're hard at work with our huge vivarium full of hundreds of overweight atherosclerotic mice awaiting injections of various approaches to therapy.
So it is possible to break down this cholesterol and the plaque itself into these local waste products, you're saying then? Yeah, is that something that's like, is that something that happens when we're younger on its own in our body? No.
What happens when we're younger is that the macrophages don't get overwhelmed to the same degree. And what they do is they ingest the contents of the plaque and then hand it off to HDL particles, which go back into the bloodstream and head to the liver where it's excreted. The problem occurs when that process breaks down because the macrophages become dysfunctional because of oxidized cholesterol or oxidized LDL particles that make them pathological. Systemic inflammation is also a bad thing because it distracts macrophages and makes them adopt their inflammatory anti-pathogen mode of operation, and that stops them doing the necessary work of ingesting cholesterol and getting it handed off. So it's a complicated little loop of cholesterol going around the body, and the macrophages are the weak point that stop working properly. So we do two things by giving them this ability to break down cholesterol. Firstly, we prevent them from getting overwhelmed because they can just get rid of the excess that makes them unhappy. And secondly, because they're not overwhelmed, they're doing their job. So in addition to breaking down more cholesterol, getting more cholesterol out of the plaque, they also continue to hand off cholesterol to HDL particles to get sent back to the liver. So they become hyper efficient of doing this very necessary job.
I see. Okay, wow, that sounds very promising. The one thing you mentioned, it's working in mice, and then you also don't have the same issues as some of the other studies do in terms of extrapolating from mice studies to human studies. I'm sure you're going to obviously do human clinical trials as well. But I am actually very curious about that. That what is, I mean, what do you think about like when a lot of people extrapolate from my studies to human studies? Like, how do you think about when it's safe to do so, when it's not? And as you mentioned, like some of your studies
Well, I
won't
mean,
have that issue.
the devil is in the details here. Sometimes it is, usually it's not. Senescent cell clearance is an example of, where it is pretty safe to extrapolate based upon what we know, in that these cells are pretty terrible. And the only question is, how terrible versus all the other things that are going on are they in mice versus people? There's no question that you're going to get benefits.
Mm-hmm.
In the case of atherosclerosis, I can talk in a little more detail as to why some things are very confusing about the reason why past therapies haven't worked. So this reverse cholesterol transport business, where your macrophages are digging cholesterol out of the blood vessel walls and giving it to HDL particles. It's a multi-step process. There's the part where the macrophage ingests the cholesterol through surface receptors. There's the part where it hands it off via other surface receptors and transporters to the HDL particles, and then there's the HDL particles themselves and their take up in the liver. So this system can be improved. You can add more HDL particles. You can upregulate the genes that are involved in the protein that produce the proteins that hand particles off or ingest cholesterol. And if you do any of this, it works really well in mice. You can upregulate any part of reverse cholesterol transport in mice, and you get reversal of atherosclerotic lesions. Some of the studies have produced results that are as good as our results, just by improving reverse cholesterol transport in mice through any one of these mechanisms. I think there have been 3 or 4 trials in humans now where they attempted to bring one of these mechanisms into a human study, and they all failed. So there's something we really don't understand about what the long poles are, or what the rate limiting steps are in reverse cholesterol transport in humans versus mice. And something about that means that anybody who tries to bring a therapy based on improving reverse cholesterol transport is you're entering the zone of death there. Many have gone before you and failed. So it's a tough one, because in principle that should absolutely work. The whole system is based on a failure of reverse cholesterol transport producing atherosclerosis. So why is it that upregulating it in all the ways that have been tried so far works great in mice, doesn't work in humans? Who knows? And this is one example of any number of examples we could point to in which something worked really well in mice and then failed horribly in humans. I mean, the Alzheimer's field is another one. And there, at least, you can point to the fact that the major problem is that mice don't get atherosclerosis. Don't get Alzheimer's in any way, shape, or form. In fact, no other species, with the possible exception of chimpanzees and dolphins, gets anything close to human Alzheimer's. So you have to produce these very artificial models, and the model is led by the assumption. The researcher says, well, I think the problem is X, too much of this protein. So then they create a mouse that creates too much of that protein, and then they fix the mouse. But, does that mean that that's going to help with humans? Because you're testing a hypothesis there. The hypothesis, you built a mouse that conforms to your hypothesis about how the disease works. And this is how we get to the point of, 15 years of work on clearing amyloid beta in the brain, leading to clinical trials in which we successfully clear amyloid beta from the human brain, and patients don't improve. So something is wrong with the hypothesis. And that field is a troubled one, because we don't have any animals that actually really legitimately have Alzheimer's disease. You're always going to have this step where somebody proposes something, then creates a mouse, then fixes the mouse, and this is a 10-year cycle of 5 years of making mice and 5 years of trying to get it into humans. And if it doesn't work, you say, well, I didn't have it right that time.
Right.
Not good.
Is there a better, faster way of even doing it, though? Or, I mean, is the only solution is to go from mice to human studies?
No, I think like the better solution is to fix the damage. Take the SENS list of forms of damage and go fix it. For example, try to clear senescent cells in Alzheimer's patients and see what happens, because senescent cells create neuroinflammation. Neuroinflammation is probably important in Alzheimer's. Did that do anything? I think that's almost a better way forward than this business of let's fixate on one thing and produce highly artificial animal models, and then go see where we go from there.
Yeah, that's an interesting point, right? Because for Alzheimer's, for example, clearing out amyloid beta plaque works in animals and mice, but then for humans, you're saying it's not as effective, and that could also be perhaps because there are all these other processes that are happening and breaking down in your body, like senescent cells, for example. I'm just conjecturing,
Yes.
but I'm sure there's like multiple. You said it's like we're such complex organisms.
Well, the end stage of aging is really complicated, and the characteristic of aging and research into late stages of aging is that it's next to impossible to determine whether A causes B or B causes A, or they're mutually interacting. All you can do is try to fix one of these things and see what happens. So that's exactly what they did with amyloid, and I think it's now being conclusively proven that amyloid is just not that important in later stage Alzheimer's disease. It might be important in the creation of the early stages of Alzheimer's disease, but I think it's probably going to take 10 years to figure out whether that's true or not, because it's a very long buildup over a very long period of time. And it may just be that amyloid is a complete side effect, that it's just something that happens because people have persistent viral infections, and amyloid beta is an antimicrobial peptide, and or it's some other consequence of inflammation that people have, and or it's not the amyloid you see, which is outside cells, but rather that amyloid is degrading the ability of the amyloid that's necessary inside cells to do its job, which was another. It's very complicated, and people will keep hacking away at this, but I do think that trying to address the deep causes of aging is more likely to get you somewhere in a shorter period of time, because you're aiming at restoration of youthful homeostasis of cellular processes by getting rid of the damage.
Okay, so discussing just addressing these root causes of aging, you also mentioned tyranny of funding in the beginning. I think one of the things is that there's just less funding probably available for rejuvenation therapies because people don't know if they're going to work in the future. Whereas maybe some of the causes for slowing aging might end up getting more funding, but probably should be the other way around. What do you think is there a narrative that you think could solve the problem? What is like a potential solution to getting more funding for rejuvenation?
Well, I think the history of this is pretty weird. It's very human, but definitely weird. So in the 1970s, a bunch of people became convinced that we could do something meaningful about aging sometime soon. And this being the 1970s, they were very focused on hormones and supplements. And they were completely wrong about what could be done and how and in what time frame. But they did successfully found an entire industry, which goes to show that business success is in no way conditional on actually meeting your goals. So we have the anti-aging industry, spawned in the 1970s, selling potions and hope and whatever. And the more effective people at selling were obviously the ones who believed their own hype. And the scientific community looked at this and said, "Oh God," and just kept it all at arm's length. And so you have a sort of 20-year dynamic in which scientists became ever more unwilling to say anything about intervening in the aging process, or even working towards that direction, or doing anything other than sort of gently examining what happened in old people. It's wasted decades until the 90s, when people were starting to show that single gene mutations in short-lived species can produce quite startling changes in the aging process and the duration of life. And that work gave rise to most of what's going on at present. But that's all slowing aging. It's calorie restriction related research. So the fight of the early 2000s was convincing the world that we could actually do something about aging and that it was worth funding. And that was a bootstrapping approach of persuasion and getting some data and getting somewhere. And then Senolytics were the first real game-changing item to come out of that. And as soon as that started to happen, then the landslide began, and now the convincing is done. People understand that we can do something about the people that matter, understand that we can do something about aging. The rest of the world still has to catch up, but everybody who's involved in funding and important decisions understands that we can potentially do something about aging now. The fight now is to make sure that the funding and the work goes towards things that can, in principle, produce large gains, which is, we want to be working on methods of rejuvenation, not methods of building a better form of exercise. But, I'd say a good 80% of the industry or more is basically working on upregulation of stress responses, things that can't change the shape of a human life, even in principle. Very trivial drugs that may do just a little bit of good. And anything like mTOR inhibitors, heat shock protein manipulation, all the rest of this stuff that's going to make you have more autophagy in your cells. It's not worth the time and money being put into it. It really isn't. It's not going to change. It's not going to move the needle. You're not going to live that much longer because of it. We need better and more than that. And that's the current fight. We need, the Senolytics industry to be repeated for all the other aspects of the SENS pipeline of ways to address the root causes of aging. Because Senolytics are really impressive in mice. I mean, really impressive compared to everything else. You can't reverse cardiac hypertrophy through any other methodology. You can't reverse kidney fibrosis. You can't make these mice appear radically younger at old age by any of these other methods. So we need more of that. If Senolytics managed that, then the other SENS items should be equally interesting.
No, I absolutely agree with you. What is a good way to fight that fight?
Well, if we knew how to make advocacy work, I'd write a book and I'd be famous for that. It's a hard problem. I mean, everybody knows there's a tipping point. But how do you reach the tipping point of getting 10% of the world to agree with your proposition? And that's really tough. And I don't know. You just go out there, you write, you talk to people, you try to bootstrap enough funding to get your to show examples that prove your proposition, and it's just a never-ending fight.
I think one thing you mentioned is that just making it, although it probably just takes just a small portion of the population to fund, and most of the people who matter, say, may have already been convinced, but I think still making it something which is more of a popular ordeal such that larger amounts of people get behind it. Because I think, as you said, people want to donate to popular causes. Hopefully that helps too.
Yeah. So there's a sort of there's a wave approach where you need a certain amount of the public to be behind your position and show they're behind your position by donating to you, because the very conservative high net worth individuals and other organizations that control large amounts of funding, they don't do things unless the path has been blazed already. Unless you have social proof, as it were, tens of thousands of people trumpeting your cause and donating to your foundation or whatever, you're not going to see high net worth people starting to support that. So it's always a case of you need the grassroots in order to legitimize the organizations that will actually provide the vast amount of funding to provide that funding. There's a power law distribution of funding for research and investment, but unless you have the lower part of that graph, unless you have the people who are donating $10 here and $10 there, unless you have them in large numbers, you're not going to find billionaires coming in to say, this is really interesting, because these people are all very, very, very conservative.
Right. Yeah, no, I totally agree with you. And I mean, it makes so much rational sense to me, especially when you laid out that, aging is something that costs trillions to the society and so much suffering. I mean, more suffering than probably the worst sort of suffering in a third world country. Uh, but I guess there's this disconnect between the broader public and the aging community, and looking at it this way, that aging is actually an important problem to solve. And I almost wonder if like we're doing a bad job in explaining this, if we can probably come up with a better explanation or narrative to help people understand why this is an important problem to solve and donate to and invest money in.
Right. And what is that narrative? That's the question. And how
Yeah.
somebody will come up with something to succeed. And the more people we have trying things, the more likely it is that someone will come up with something that works really well. But ultimately, if you look at all causes, even the seemingly fast ones took a lot of time. I mean, AIDS advocacy was an uphill battle for a long time until the world decided that, oh yeah, actually maybe we should be doing something about this. And the man in the street understands that, yeah, we donate to AIDS research because it's the right thing to do. And the same thing was true of cancer research. It took like 20 or 30 years to get to the point of people collecting for cancer research in the street, and understanding that, yeah, we should fund cancer research. So I think we still have a way to go, maybe a decade or two, before people understand that, yeah, actually this is the thing we do. We try to fight aging. Right now, we're still at the stage of the people who control funding and the people who set research agendas are sort of on the team, and are really just arguing over how to go about it. And everybody else is going to catch up at some point. But can we do that faster? And would it matter? Faster is always better, because there's no amount of money that could be too much to put into this work. There's so much, the cost is so enormous, and the amount of work to be done is so huge. So we could multiply the research community by a factor of 10, I think, and that still probably wouldn't be enough to make the sort of progress we'd like. So clearly there's a lot of work to do. How do you make it faster? I wish I knew.
Yeah, I'm hopeful. I think there's progress being made. I want to end with 3 quick questions. If hopefully someone has been convinced to invest or donate this money in this field, where are some places they can do so?
I think, like, in terms of donation, you can go straight to the SENS Research Foundation or the Methuselah Foundation. And if you want to donate a larger amount, you can go to them and not just donate, but you can actually have a conversation and say, okay, what are the projects you're working on? What are the projects you'd like to work on? And see if you can find something that interests you and that you understand. That's always a possibility. In terms of investment, I think the best way to go about that is go to the conferences. Obviously, that's terrible advice for the last 2 years because we've been having a pandemic. But now that conferences are starting up again, there's a number of conferences, such as Ending Age-Related Diseases in New York. There's Longevity Therapeutics every year in SF. There's Undoing Aging, which is in Berlin these days. And these are all, there's probably a couple others that don't spring to mind immediately, but these are conferences with a really good mix of scientists, entrepreneurs, and investors. And if you go there and start talking to people, you'll get a sense of what the informal groupings are in the angel investor communities and who's who. And that's really the only way to do it, because investing it's a network thing. And it's very resistant to putting itself online and places where you can find it. It's all in person and who and that's really the way to do it. You have to figure out, what's the network here, get introduced to people, and then go from there.
Yeah, totally agree with you. I come from an investment background. So second question, what are some longevity protocols you follow right now, I guess? I mean, apart from exercise, caloric restriction, are you doing Senolytics or anything else, any other supplements?
So I publish as a self-experimenter, I publish protocols every now and again at Fight Aging. There's a self-experimentation section buried in there somewhere, sort of the lists, the ones that I've thought about or tried. So Senolytics, yes, absolutely. I take those. sort of once every 6 months or once every 3 months. I've done flagellin immunization, because that has pretty good results on restoring the gut microbiome to a more useful level. And that's one where, who knows what the outcome will be in terms of benefits to health, but it's something where you can really easily measure it. And sometimes that's more interesting and useful from a self-experimentation perspective. There's a few others that I've looked at and indulged, and of course I've tried NAD plus upregulation and not seen anything tremendous coming out of that because it's a small effect. But self-experimentation.
How do you measure that?
Well, in theory, you would look at things such as you go do blood work, look at your inflammatory markers. You measure your endurance. You would look at cardiovascular measures such as resting heart rate variability, pulse wave velocity, which are all very hard to measure consistently, by the way. Unless you're very old, you're unlikely to see measurable benefits from things like NAD plus upregulation. But people who are very old in their 70s and 80s have seen quite impressive results from things like NAD plus upregulation, exosome transplant, stem cell therapies. There was a fellow at one of these conferences who was in his 80s and walking with a walking frame, but the point was that he was walking even with a walking frame, and he couldn't do that before he'd gone and had one of these mesenchymal stem cell therapies, which are actually pretty good at addressing chronic inflammation. And chronic inflammation, of course, is a pretty sizable component of frailty. If you're very old, a lot of these things that I shrug over are actually worth looking at because there is some evidence for them to have general benefits in your quality of life.
I see. Okay. And lastly, is there anything else that you would like to discuss we may have missed, or anything that you're seeking help on if someone watching this could help?
Well, I'm always interested in talking to investors. Obviously, my company raises funding, and we're raising funding right now. So if you're an angel or a small fund, family office, come talk to us, Repair Biotechnologies. Beyond that, there's always something you can do if you feel that you actually do want to try to make a difference in this field. There are always nonprofits looking for help. There are increasing numbers of people forming institutes and undertaking studies and helping researchers do their thing or investing in companies. Come turn up to one of the conferences if you're interested in participating, and see what you find in terms of things you could help with or that you'd like to do.
That's great. I certainly hope more people get involved.
Indeed. Thank you.
Well, thank you so much for your time, Reason. This has been great and very informative.
Okay, you're welcome.
Thank you.
Hi, everyone. If you enjoyed today's episode and learned from it, I would love it if you could subscribe to the channel and like the video. It helps get the word out. Additionally, it would mean a lot if you could show some support and share the channel with your friends and family. I want to get the word out on aging and longevity science to as many people as possible, such that we can all be healthier for longer. Okay, if you enjoyed this content on longevity science and science-backed health optimization and longevity lifestyle hacks, you can check out my newsletter, which is livelongerworld.substack.com. You can also listen to the audio version of this podcast on Spotify or other places that you get your podcast if that's the preferred medium. You will find all these resources on my website, which is livelongerworld.com. All right, I do have social media as well. So if you want to follow me on Twitter, I am at livelongerworld. Instagram, I am at Longevity Future, where I often post graphics distilling the basics of longevity science and also talking about longevity lifestyle hacks that I personally practice. Lastly, you can support my work by donating on Patreon, which is simply patreon.com/livelongerworld. All of this is also included in the show notes, so if you want, check that out. And that's it for now. Stay in good health, and I will catch you next time.